Okay. So, good morning, student. In my first lecture, I discussed about the fundamental concept of fast reaction, and in second lecture, I discussed the continuous flow method, and there are some problems of that continuous flow method. Number one problem is that large quantities of reactants are required. Number two, if the half-life period of a reaction is 1 millisecond, then of course that continuous flow method will not be very suitable. So therefore, another method is developed which is very similar to continuous flow method, that is called the stopped flow method, and it is a very popular method because you can study any reaction whose half-life is less than 1 millisecond. That means suppose 1 into 10 to the power minus 3 seconds. Number two, that very small amount of reactants are required, and theory is almost same as the continuous flow method. The only difference is that we have to stop that reaction. Okay. Now, that continuous flow method was developed by two great scientists, Prof and Rutherford, but this method was developed by a great scientist, Professor Brighton Chance, and he developed this method in 1940, and he published large number of research papers on this. He used different types of fast reactions. Basically, most of the biological reactions are very fast, like enzyme kinetics, or sometimes protein-protein interaction type of things, ligand binding reactions, or coordination type of the reaction. That type of reaction can be studied with the help of this stop flow method. So this is the first paper. Title is the accelerated flow method for the rapid reactions, and it is published in Journal of Franklin Institute. Not only that, he also published different types of method, and this is their second paper in 1942. You see that he studied some of the enzyme substrate reactions, and I will show you a device that actually what we need a separate type of the device, so that both the reactants are forced to reach in a mixing chamber, and one reaction is completed. Then of course, with the help of any spectrophotometric method, we can monitor that particular property. I am just showing you, this is also another very good paper, of course written by the other scientists, and there are slight modifications are also there, and still people are using different types of reactions. That means only thing is that you need a device. Suppose I am just showing you that one device. This is actually one type of device, and I'll explain each and everything of that reactions. I will explain each and everything of that reactions, because suppose you are using simple reactions, so always you need two reaction vessels, and mixing time at least 0.5 seconds or 0.5 minutes. During the mixing, reaction is occurred. So you have to minimize that mixing time. So that is a special type of device is required. And if you see very carefully this instrument, because I am grateful to Defense Research Development Organization, DRDO, Government of India, and I think 2005, today is 2023. So I think 18 years ago, I purchased this equipment, this is applied photophysics, for studying some of the fast reactions, 2005. So, just I explain you things. It is neither spectrophotometer nor a spectrophotometer, not spectrophotometer. Only thing is that it is a device, and there are two syringes. One is called reactant A, another is called the reactant B. Just like you just imagine a two beaker or two reaction vessels. And there is a mixing chamber is also there. And you see, this is called umbilical cord, because there are two reasons. First, it is an automatic device. There is a pump system is also there. Once the reactants are ready, everything is ready, but we have not mixed it. There are two separate things, and there is another part, that is the mixing chamber, and automatically when I just press this button, that reaction is stopped. And this umbilical cord is connected with any type of instrument. I will show you the picture. Suppose you are having a UV visible spectrophotometer. Suppose you are having a fluorescence spectrophotometer. Suppose you are having a FTIR. So if it is a UV visible spectrophotometer, what you will do? You will monitor the absorbance. If you have a spectrophotometer, so you will monitor the fluorescence intensity like that. And this umbilical cord, I will show you that it is connected with that instrument. So it is a hyphenated technique. That means this is a separate device for sending the chemical reactions or the reactants into that instrument, and with a fraction of second, it is automatic system, and then whatever the absorbance, it may be increased, it may be decreased, monitored by the spectrophotometer. So this is the simple principle of this stop flow technique. Now, why this point is important, that yesterday also I discussed with you, everything is depends upon the half-life period. Because if the reaction is 1 into 10 to the power 3 like that, then of course flow method is suitable. Tomorrow I will explain the relaxation technique, and relaxation technique range is 10 to the power 6. Sometimes if the reaction rate is very high, 10 to the power 8 like that, rate constant, and of course half-life is 10 to the power minus 8 or minus 10, then of course other method will be used. Now, as I already discussed with you, that most of the biological reactions can be studied with the help of the stop flow technique. And we actually, during this DRDO project, we studied the protonation studies. And protonation study, suppose I am taking a it is a analytical reagent, and if I add some acid, so there is a very fast reaction, maybe nitrogen protonation species or oxygen protonation species. So it is called the protonation studies, S plus H, SH, very fast, K, SH. So we use this method. So it can be clubbed with the NMR, it can be clubbed with the UV visible or anything. NMR is the most suitable method because proton can be. I'll show some of the picture of all these things. And you know that electron transfer reaction, I think 4th semester you will study the electron transfer reaction. So biological reactions, then coordination reaction, ferric and HCN reactions, protein folding type of the reaction can be studied with the help of this type of the reaction. Now I am coming to some block diagram. Now there are two important points is there. One is called the deadline, dead time, and to avoid the mixing time. So that is the difference between the conventional method and technical method. That in conventional method you can't avoid this mixing time because it is a manual method. Whereas here we need say very, it is 18 years old instrument. Now everything has been changed. Now very sophisticated equipment is also there. So complete mixing time can be avoided automatically with the help of digital system that both the solutions are forced to go this thing. And just after steady state we stop the system with the help of this thing, just to stop the system. And there is a drainage system also because after this reaction, so we inject that solutions. That is also there. So the flow is stopped suddenly with the help of some electronic device, and measurements are made spectrophotometrically of the concentration of the function of time. That is just like a normal system. So if I am taking some para-nitrophenyl acetate, I am just giving one example. And kinetics is, there is no difference between the basic theory of the kinetics. Either you monitor the concentration of the reactant or you monitor the concentration of that product. And here product is para-nitrophenoxide ion, dark yellow color. And you are using different types of alpha nucleophile. So immediately the concentration, if the reaction goes on, so very fast, para-nitrophenoxide ion and slightly disease, complete reaction. Sometimes what happens, suppose you are taking some other type of the reactants where concentration is decreased, so you will get also such type of things. So it is depends upon the technique. Either you monitor the concentration of the product or monitor the concentration of the reactant with respect to time. Only millisecond, microsecond like that. Millisecond, microsecond, nanosecond, picosecond. So kinetics, there is no change. Only thing is that we need a device and there must be, everything is now computerized. If you visit to my lab, you will see in spectrophotometer there is a computer, automatic. There is a fluorescence, there is a computer. For surface tension, there is a computer. So now everything is, but long back, there is no computer. So only thing is that the automated, that manual system. Now everything, even if you go to the NMR, there may be magnet, but it is controlled by a small computer. Everything is computerized. You need not be worried about how we monitor this, their properties like that. So two reagents are rapidly mixed. It's just like an injection. I told them to bring that. So there are two, like very small size of injection series. So rapidly mixed together and then stopped in the observation cell. You see there is the observation mixing chamber. You will see there that color also. And sometimes they have given an additional system. Suppose A plus B plus C, as I told you. So there is additional. So A plus B plus C. That is also possible. As the reaction proceeds, the change in fluorescence or absorbance at a specific wavelength is recorded as a function of time. So only thing is that it is a device. Many times I am telling something, nothing new. You must understand. So just prior to the stopping, a steady state flow is achieved. The solution entering the flow cell is only millisecond old. Now, dead time. The time between the end of mixing and the start of the measurement typically in millisecond range. But just you just think about the hydrolysis of methyl acetate, mixing time at least 1 minute, that is the dead time. Time between the end of mixing, keep it out and just so and start the reaction. That can be avoided with that can be solved with the help of such. Therefore, it is a stop flow technique. In normal system, you are using ice cold water for hydrolysis of methyl acetate. Just quench the reaction. Otherwise, how will you measure the rate constant? In some cases, you boil the reaction, hydrolysis of methyl acetate, for complete with that reaction. So that is called the quencher. Here no quencher, you just stop by the method. Now this is the most simple diagram, block diagram, and I am always, as I told you, that I always preferred the Ledler book. And this is the most important diagram. You can understand this diagram, that this is the type of two reservoir, red color. You cannot see these things. Just if you can these things. And then there are, why it is reservoir? Because before the reaction you just, but this is actually syringe. Two color I can write. Automatically it will enter these things. One is red, suppose this is A. So these two things, A and B, and this is actually starting point. Then there is a system is also there. It is not there. We can push it up. Just to open it. So you can show the inside. That is actually the red and green color derived syringe. Two syringes are there. This is reservoir and here two syringes. And this there is a mixing chamber. This is a mixing chamber. because this is a block diagram. And constant temperature bath. We just simply we connected with a Peltier thermostatic bath. Suppose you would like to conduct the experiment at room temperature or suppose 30 degrees centigrade. So there is a separate device that is called the thermostat, automatic thermostat. So we circulated that water. So there are two inlet, outlet. So it is water is automatically circulated. And drain bulb block. After that reaction, we pour this reaction to drain block. And here only thing is that there is an umbilical cord. That cord with a cuvette. This is a cuvette. So that umbilical cord is actually connected with that instrument. I will show you that picture. So this is a schematic diagram of this top flow apparatus. Solution in the two drive syringe A and B are forced rapidly, automatically, through a mixing jet into a cuvette. When the flow is stopped, then of course, by that method, here they are using a UV visible spectrophotometer, so photomultiplier tube is there, and there is also direct machine is also there. It's a very costly machine. So this is the actual, see the, this is this instrument I have shown you. This is called umbilical cord, you know, in biological system, umbilical cord. After the birth of a child, we just cut that. That is called the umbilical cord. So that is, this is the cord, this is cuvette. Now in first case, this is our carry 60 UV visible spectrophotometer. And when I started my career as a lecturer, first I purchased a Pi Unicum. I have purchased at least 3 to 4 UV visible spectrophotometer long back, because I have been working in this department last 35 years, 36 years, and I have successfully completed 15 projects. 15 projects. So at present I am not doing any project, but all these instruments, the UGC SAP and DST FEAST also I was coordinator. So you just this cord, and we keep this apparatus in front of that instrument, like you can understand these things. That the details of that, where is that photograph where that instrument is lying with, oh, it should be, you see, it is computer automatic computer system. So we will keep this apparatus above this instrument, okay. Then we monitor these things and just put this umbilical cord inside that things. These are the details of this instrument. Yes, this is the actual position of these things. Very recently I have taken this photograph. That time there was no mobile and other things. So you see, the size is also just like your cuvette, just like your cuvette. So it is connected with this cuvette, and rest is automatic system, and this computer is monitored the systems. Then we are measuring the absorbance, optical density. Suppose I would like to measure the fluorescence, because sometimes the UV visible is not, then we use this is actually spectrofluorimeter, different. This is actually thermostat for maintaining the temperature. It is Peltier operated thermostat, and again same thing, same instrument, same thing. Only thing is that now we will measure the fluorescence intensity. So same thing, same system, nothing new. So this is connected with that instrument. So it is a hyphenated technique. So last part is that it is a volume of the reagent is very small. You can say 1 ml or 2 ml like that. And a permanent recorder can record the progress of the reaction in a wide range, absolutely no problem. But where in continuous flow we have to monitor the volume element, which is very difficult to measure accurately. But here Nothing, everything is automated, and this method is not affected by the rate and character of the flow. So, thank you.